Mosses cling to the wet earth, restricted to a two-dimensional existence. But beside them, a fern unrolls its fronds, reaching several feet into the air. These towering plants are pteridophytes. They were the first botanical structures to successfully breach the vertical limit of early terrestrial ecosystems. Growing upwards requires defying gravity to move water from the soil to the highest leaves. Pteridophytes achieved this by evolving dedicated vascular tissues. Specifically, they developed xylem to pull water up, and phloem to distribute synthesized nutrients back down. This internal plumbing places them directly on the evolutionary timeline between moisture-dependent bryophytes, like mosses, and the highly advanced seed-bearing plants that would follow. Without a specialized internal transport network, any plant attempting to grow tall in dry air would rapidly desiccate and collapse under its own weight. Engineering this internal plumbing was the absolute prerequisite for vertical growth. Intrigued, pteridophytes present a distinct biological contradiction. They possess advanced internal plumbing, yet they lack the reproductive hardware of modern plants. They produce no flowers and yield no seeds. Because of this, botanists classify them as cryptogams. This term translates directly to hidden reproduction, categorizing plants that reproduce without visible seeds or flowers. Instead of seeds, pteridophytes rely on microscopic spores to expand their territory across the land. To understand how a plant with advanced vascular tissue reproduces via simple spores, we have to look at their Haplo-Diplontic life cycle. This is a biological system where a single organism alternates between two completely separate, independent living phases. To map the life of a pteridophyte, one cannot just look at the fern. Both independent biological phases of its existence must be accounted for. The top half of this cycle represents the phase we are most familiar with. This is the sporophyte. It is the macroscopic, independent, and physically dominant body of the plant. It features true roots, an underground stem called a rhizome, and large, complex leaves. Those leaves come in two distinct architectural types across the division. Some species produce microphyls, which are small leaves with a single unbranched vein. Others produce macrophyls, the large, highly divided leaves commonly known as fronds. While their vascular tissue was an evolutionary breakthrough, it still carries primitive limitations. The water-conducting xylem lacks the highly efficient tubes known as true vessels, and the nutrient-conducting phloem lacks companion cells, which help modern plants load sugars. Despite these limitations, the sporophyte is a highly capable autotroph. It drives photosynthesis using a specific chemical payload of chlorophyll A, chlorophyll B, and carotenoids. The sporophyte is a solar collector and water pump. Its entire anatomy is optimized to capture sunlight and maximize vertical growth. The reproductive mechanisms of the sporophyte are hidden on the underside of the foliage. These specific fertile leaves are called sporophylls. Their sole biological task is to bear sporangia, the microscopic capsules where spores are manufactured. Pteridophytes organize these sporangia in different ways. Ferns group them into distinct clusters called sori on the underside of their fronds. Genera like Selaginella and Equisetum pack their sporophylls tightly together at the tips of their stems, forming dense cone-like structures called strobili. Inside each sporangium, a diploid spore mother cell, carrying two complete sets of chromosomes, undergoes the process of meiosis. This meiotic division splits the genetic material in half, resulting in the creation and ejection of 4 distinct haploid spores. The act of meiosis serves as a strict boundary line. It terminates the sporophyte's reproductive process and physically launches the plant's second phase of life into the wind. When a microscopic spore lands on dark, damp soil, it immediately begins to germinate, sprouting a green cellular filament. Through rapid mitotic cell division, this single haploid cell grows into a multicellular organism known as the haploid gametophyte. In pteridophytes, this structure is specifically called the prothallus. The prothallus is completely independent and photosynthetic, but it is structurally primitive. It lacks true roots, relying instead on simple, hair-like rhizoids to anchor itself. More importantly, it possesses zero vascular tissue. Without an internal water transport system or a protective waxy cuticle to prevent evaporation, the prothallus is highly vulnerable to drying out. This creates a strict ecological tether. The massive, towering sporophyte fern can only exist where its fragile, non-vascular prothallus can survive, restricting the entire division to damp, shaded microenvironments. To complete the life cycle, we must look closely at the surface of the prothallus. Here, the prothallus develops its sex organs, the spherical male antheridia and the flask-shaped female archegonia, which holds a single egg. Fertilization in pteridophytes is completely reliant on environmental water. They utilize zoidogamous fertilization. This means the male antheridia release multiflagellate sperm that physically require a liquid water layer to swim toward the archegonia. The sperm fuses with the egg to create a diploid zygote. This single cell immediately begins mitotic division, drawing nutrients from the prothallus to sprout a brand new, leafy sporophyte. In most ferns, this cycle relies on homospory, meaning the sporophyte produces only one identical type of spore that grows into a prothallus bearing both male and female organs. Certain genera like Selaginella and Salvinia developed a different reproductive strategy, heterospory. Heterosporous plants produce two distinct spore types. Microspores germinate into strictly male gametophytes, and much larger megaspores germinate into strictly female gametophytes. In this system, the female gametophyte does not grow on the ground. It is retained and protected within the megaspore, remaining attached to the parent sporophyte body. Retaining and protecting the female phase inside the parent plant eliminates the extreme vulnerability of the free-living prothallus. This specific mechanic is the evolutionary prototype for the modern seed. Botanists classify this wide range of vascular and reproductive experiments into 4 distinct taxonomic classes. The most primitive class is Psilopsida, which includes Psilotum. These plants lack true roots entirely and often function with simple, leafless green stems. Next is Lycopsida, commonly known as club mosses. Genera like Selaginella and Lycopodium belong here, characterized by their tiny microphyls and cone-like strobili. Sphenopsida contains the horsetails, such as Equisetum. You can recognize them by their distinct jointed, ribbed stems and whorled, scale-like leaves. Finally, Pteropsida encompasses the true ferns, like Dryopteris and Adiantum. These are the plants that dominate forest floors with their massive, complex macrophyll fronds. This 4-part framework serves as a map of early botanical experimentation with vascular tissue and leaf structures. Beyond their evolutionary history, pteridophytes maintain a highly active role in modern human and ecological systems. Ecologically, they are aggressive soil binders. Their dense, fibrous root networks grip loose soil, preventing severe erosion on steep embankments. In agriculture, the tiny aquatic fern Azolla serves a critical function. Azolla maintains a symbiotic relationship with Anabaena, a nitrogen-fixing cyanobacterium. When cultivated in paddy fields, this partnership acts as a highly effective natural biofertilizer. Their utility extends directly to humans as well. Beyond widespread use as ornamental plants, species like Dryopteris contain chemical compounds used to synthesize anthelmintic drugs, which treat parasitic worm infections. Pteridophytes function as active structural and chemical participants in our modern ecosystems and industries. The Pteridophyte division functioned as the primary laboratory for terrestrial botany. To survive in dry air and gravity, they engineered internal vascular plumbing. To protect their offspring, heterosporous species developed specialized spores that kept the female gametophyte attached to the parent plant. By solving these physical limitations, the seedless cryptogams laid the groundwork for the seed-bearing phanerogams that followed them. The mechanics of internal water transport and heterospory provided the exact architectural blueprint that gymnosperms and angiosperms would use to eventually colonize the globe.